Thanks for your prompt on-time return. We're ready to continue. It's my pleasure to welcome Dr. Tabras Siddiqui, Professor of Physiology and Pathophysiology, and a PI at the Clayson Center for Advanced Medicine. Tabras.
Good morning, everyone. Thank you for being here. It is my pleasure to introduce Dr. Sheena Josselyn. She's a senior scientist at the Hospital for Sick Children and a professor in the departments of psychology and physiology at the University of Toronto. She holds a Canada Research Chair in Brain Mechanisms Underlying Memory and is a Fellow of the Royal Society of Canada. She earned undergraduate degrees in psychology and life sciences and a master's degree in clinical psychology at Queen's University. And then she went on to do her PhD in neuroscience and psychology from the University of Toronto and conducted postdoctoral work at both Yale and UCLA. And UCLA, I think you were with Alcino Silva. Dr. Josselyn's awards include the Innovations in Psychopharmacology Award from the Canadian College of Neuropsychopharmacology, and the Efren Award from the American College of Neuropsychopharmacology, and the Andrew Carnegie Prize in Mind and Brain Sciences. But the award that she is most proud of is being the class president in 4th grade. Dr. Josselyn is interested in understanding how the brain encodes, stores, and uses information. Specifically, her lab investigates how specific neurons are recruited or allocated to an engram, and how neuronal membership in an engram may change over time or with new experience, some of which she will talk to us about today. She lists in her Twitter bio, that she is a short Canadian, but she is in fact a giant in Canadian neuroscience, and we are very, very pleased to have her among us today. Sheena.
Well, thanks so much for that lovely introduction. And yes, we all have our own trajectories, and I just managed to peak in the 4th grade, so downhill from there. So I'm really excited to be talking to you today. I think this is a great event. It's nice to see everybody in person. And although I had a wonderful introduction, I'm going to also give you a little bit more detail about me. So who am I? First, I'm a proud Métis. I'm a member of the Métis Nation of Ontario, so shout out to representation. I am also an American. I know, born in Cleveland, accident of birth, but now Canadian. So my family moved to Kingston, and like Ellen, I went to Queen's University in Kingston for my undergrad degrees, master's degree in clinical psychology, on to University of Toronto. And then I did several postdocs. So the first thing I did was a clinical postdoc where I sort of thought I was Jodie Foster in Silence of the Lambs. I was Clarice. I worked with sex offenders in prisons in Ontario. And I thought that I would come and sort of match wits with these evil geniuses. And I did this for about a year, and then I realized that they weren't really evil geniuses. They were just very, very mixed up, sort of weird people, and that I really longed to get back to real science. So then I did postdocs at Yale and UCLA. And fun fact, while I was at UCLA in the neuroscience department, I ran into, I don't know if you know her from The Big Bang Theory, she was getting her PhD in neuroscience at the same time. So it was sort of interesting that she went on to other things, and I stayed in science. And then in 2003, I joined Sick Kids, and this is our lovely research building, and my lab is, around there. So this is who I am. Now, what do I do? My lab studies memory. We try and study how the brain codes, stores, and uses information. We think this is a real fundamental process in science. Understanding this, we think, will open doors to many understanding of many human disorders that the root cause of which might be problems in processing information. So you might know this person. This is Alzheimer. And of course, there was a disease named after him, but the disease should probably be named after the patient that he diagnosed. Here's the patient that he diagnosed who came to be treated by him by saying, "I have lost myself," so to say. So what do we do when someone has lost themselves, when somebody has a memory disruption? Well, really similar to how we think about a car. In order to fix it, when it breaks, we need to know how it normally works. So we really need to understand how the brain normally works, how we normally store information. So really similar to when somebody has a heart attack, we're going to say, okay, we need to know how the heart works. And we have a pretty good idea about how the heart works. So it's no casting aspersions on other areas of science, but the heart is pretty easy. It's a pump. If I have problems with my heart, we can replace my heart with a pig's heart. If I have problems with my brain, we'll just leave it there. So what my lab is trying to do is to understand the human brain. But a lot of the things that we do just can't be done in the human brain. So we study the next best proxy, in our opinion, which is a mouse brain. And we're not really interested in sort of understanding what makes mice tick. We really want to understand fundamental processes so that we and other groups can then apply it to humans. So we study memory. And the idea that memory is stored as some sort of enduring change, maybe in the brain, dates back to Plato and Aristotle. I don't know which is which, but I'm told that these are those famous, very famous people. I'm going to show you a picture of who I think should be up there with Plato and Aristotle, and his name is Richard Wolfgang Semon. Now, Richard Wolfgang Semon was a German evolutionary biologist, much in the vein of famous evolutionary biologists. He went on excursions like Darwin did, and he even found a species of lizard that still bears his name. Now, I am not an evolutionary biologist. I follow Richard Semon because he became a real memory philosopher, a real sort of theoretician about memory. So why did he do this? Well, interesting story. He got into a lot of trouble. He ended up having an affair with the wife of the president of his university. He ended up losing all his funding, getting kicked out of the university system, and then he went off to do his theorizing about memory. He wrote 2 very, very influential books, and he's the one that came up with this term I'm going to be using today, engram. So he defined it as the enduring, though primary, latent modification in the irritable substance, the brain, produced by a stimulus. So what is it? It's basically a memory trace, that bit of a brain that seems to store a memory. And he also came up with another important term, ekphory, which is basically memory retrieval, which he defined as the influences which awaken an engram out of its latent state into one of manifested activity. So Richard Semon, you might not know his name. His work was very much overlooked during his life. He ended up becoming very, very depressed and took his own life in 1918. And his work was very overlooked by a lot of generations of memory researchers. But there's been a bit of a renaissance in reading back and going back and looking at his work, because he really has foreshadowed a lot of what's going on in neuroscience today. So here's my next picture. And this is me, and this is Paul Franklin and Stefan Kurler. And we met in graduate school at University of Toronto. I married Paul Franklin. He's still my husband, and Stefan wouldn't marry me, but we are still very close collaborators. And what's really cool is that the people that you meet in graduate school, many of you are in graduate school, become your colleagues for life. They are the people that you can say, I never really understood what that chromatin thing really was about. And they're the ones that will set you straight, and you can always rely on them. So they become your colleagues for life. So this is a picture of us way back in the 1990s sometime, where it's very sunny out, we're all wearing shades, and we continue to collaborate. So every few years, we get together and write a review. So Stefan is a human memory researcher. I study memory in mice, and as does my husband, Paul. And we ended up sort of having a very deep discussion at one point about Richard Semon and how he made these amazing contributions, we thought, to the field of memory, and how he's been so overlooked. So we ended up going back and saying, okay, let's look back in the literature and trace out the people that are really important to our thinking, and we called it Heroes of the Engram. And we went through and we profiled a bunch of different people, and we challenged people to say, okay, let's go back and read some history, and why don't you sort of come up with your own heroes in your own field? So we came up with a bunch of different people that were really, really important to our thinking about memories. Started out with Richard Semon, Karl Lashley, Donald Hebb, Wilder Penfield, Brenda Milner, James McConnell, and Richard Thompson. And when we were writing this paper back in 2017, we were thinking, oh, this is so cool. These people are so influential to how we think about memory today. But then we said, you know, what's really interesting is that none of them will ever win a Nobel Prize. Now, why is that? Well, the majority of them were dead at the time. And one of the written rules of the Nobels is that it can't be awarded posthumously. Now, Brenda Milner is still very much alive at over 100 years, still at the MNI. But she violated the second unwritten rule of the Nobels. Yes, she's a female. Let's look at that a little bit more. So if we look at the who's received Nobels in sort of medical sciences, physiology or medicine, chemistry and physics, we have over 600 Nobels being awarded since 1901, and only 23 have gone to women. And if we look at other historically excluded groups from science, the numbers are going to be even lower. So it's about time we start recognizing the contributions of this diverse population of people. I am happy to point out, though, that Brenda Milner, along with Endel Tulving, was awarded a Gairdner in 2005. Gairdner is always the forerunner of these things. So let me talk a little bit more about some of my other engram heroes. So this is Karl Lashley. Now, if you've ever taken Intro to Psychology, Psych 101, you might know his name. So he was an American physiologist who searched for an engram, that bit of the brain that stores a memory. And he ran a bunch of different experiments. I'm just going to show you one that he did here. He used rats, and he trained this rat to traverse this maze to find a bit of cheese at the end. And this is actually one of the mazes that Karl Lashley used. So he said, okay, this rat knows this. This rat has this very good memory. Now I'm going to go in the brain and try and find exactly where this memory is stored. So he systematically lesioned out different bits of the cortex, trying to find exactly where this memory was stored. And he did this experiment and variations on this experiment for about 30 years of his career. And then he published a very important paper called In Search of the Engram, where he basically concluded the engram is everywhere in the cortex, but nowhere specifically. He could not find an engram, and he called it elusive. And from that point on, studies trying to find the sort of biological basis of an engram or a memory trace in the brain, at least at the sort of cellular neuronal ensemble level, were largely abandoned in 1950. But I am forever hopeful, and when I came into the field, I published this paper, sort of a cheeky title, Continuing the Search for the Engram, Examining Mechanisms of Fear Memories. Because I thought maybe there was some things that Lashley did right. I wanted to build on them, but change them up just a little bit. So I thought, well, maybe the task he's using is really overly complicated. So he would train rats, to find this bit of cheese through this maze, and we don't exactly know when the learning occurs, and maybe the rats knew this too well, and maybe it'd be really hard to get rid of this memory. So I decided to use a very simple type of Pavlovian conditioning. Pavlov, he would play a tone and give dogs food, and play tone, food, and soon enough the dog would start to salivate when they heard the tone. Well, I did something sort of similar to that, only I used mice, and I used auditory fear conditioning. So I would place mice in a unique place, I would play a tone and give it a single foot shock. So not enough to cause any damage to the mouse, very much like when you walk on a carpet and you touch a door handle, that sort of shock. So enough to say what the, but not to do any sort of harm. But the cool thing about this task is that I can ask the mouse anytime later on by simply playing the tone. Do you remember the last time you heard this tone, you got this kind of aversive foot shock? So what do I look for? How do I know that the mouse remembers this? Well, you can assess fear and other emotions really, really easily in people. So this is a picture of my daughter Charlotte a few years ago wearing some sort of wacky Halloween costume. And you can just look at her right here. She's fearful. You don't have to ask her. You just know by looking at her face. And here she's really bored and wants to stop talking to mommy and play on her iPad. We do the exact same experiment in mice, and it's really hard to tell. This mouse is fearful, and this mouse is bored and wants to play on the iPad. So what are we going to do? So most of my job is trying to ask the mouse a question that the mouse can answer. And when mice are afraid, they assume this crouched motionless posture called freezing. If you've ever seen a mouse in the wild or in your kitchen or something, and you startle it, they freeze first and then they run away. So we're going to always measure freezing as our index of how well this mouse remembers this memory. So we're going to do this because it's a quick one-trial sort of learning. A mouse can remember this for their entire life, 2 years in the lab. And the training takes 5 minutes, the testing takes 5 minutes, and it's all very automated. Plus, we know where in the brain to start to really look for this engram or memory trace. So Lashley looked all over bits of the cortex, but a bunch of researchers have shown us that we should really be looking for this type of memory in this area here, this blue area called the lateral nucleus of the amygdala. So this is a see-through mouse brain. Here's the front of the mouse. Here's the back of the mouse brain. Here's a coronal, so an ear-to-ear section. And we want to look just in this location right here, because previous research has suggested this is going to be a real key area. So what are we going to do? Well, we're going to harness the power of viruses and genetic ways of manipulating the activity or the properties of individual neurons. So when we started this work, we started off using replication defective herpes simplex viral vectors, but you can use any sort of viral vector, and it's really an easy way of manipulating the different properties of individual cells. So we chose herpes because when we first microinjected it into the brains of mice, we infected about 10 to 15% of these large pyramidal excitatory neurons in the lateral nucleus. Here's an outline, and it seemed to infect cells pretty randomly. So what did we do? Well, we're going to have a group of mice, and we're going to inject our viral vectors. And the first time we did this, we overexpress a transcription factor called CREB that work in aplysia, work in fruit flies, has shown us has been really, really important in memory. So we're going to randomly increase CREB levels in a random population of cells, and then we trained mice on this auditory fear conditioning task, and we found that memory was enhanced, which is pretty cool. There's many different things you can do to decrease a memory, but to increase a memory is pretty cool. And not only that, but we found these neurons that had increased levels of CREB, that they were more likely to be active following memory recall. So we thought that this biased their allocation into the engram supporting this memory. So we are very excited about these findings, but it's all pretty correlational. You know, we saw an enhancement of memory, that's cool, and we saw this overlap of activity and these and in neurons that we had increased CREB expression in. So the real cool experiment would be to say, okay, if we think these neurons overexpressing CREB are really key to this engram, really key to storing this memory, the trick would be to go in and kill just these cells after a mouse has learned something and see if the memory is gone. So this is sort of an update of the Lashley experiment using more modern tools. So here we're going to use molecular and genetic tricks to kill cells to try and erase a specific memory. So we have two groups of mice. One group, we're going to have viral vectors that express the transcription factor CREB, as well as GFP, so we know where these, which cells are infected. And we're going to have another group of mice that just overexpress GFP. Doesn't do anything to the cell, but we infect a similar number of cells. So once we have that, we're going to train the mice. And then we're going to use some genetic tricks that Janet is pretty familiar with to kill just those cells that we've infected. So cells either expressing CREB that we think are really important in the memory, or another group, we're going to kill the same number of cells, but cells that aren't, we don't think, really important in the memory trace. And what happened? Well, we were able to ablate this specific memory by deleting this small population of cells. So only in this one group did we see a memory deficit. We didn't see a memory deficit in the other group. So we were very, very excited about this. The postdoc in my lab who did this work, Jin Hee Han, was super excited. And I remember the exact date when this paper was finally coming out. So we're like, oh, this is so cool. We've erased a memory in the brain. That is so neat. And this happened way back, March 8th, 2009. Well, how do I remember that so well? I'm a memory researcher, but I have a really bad memory. How do I know that? Well, this is me at the time. So this is me, March 7th, 2009, and I am fully pregnant here. And look at me, I'm still wearing a Kids Science t-shirt. Always wearing the colors. And this, I'm across the street, across the street from Sick Kids at Mount Sinai, and I am about to give birth. And the postdoc in my lab who'd done all this fantastic work wasn't comfortable talking about it. He thought that his English wasn't good, whatever, but his English was great. So he wanted me to do some press for this. But of course, you know, timing, life, things didn't work out so well. So this is me trying to write some things for different press accounts, but eventually I had to put all that away and have an emergency C-section. But the next day, front cover, Toronto Star, do you remember newspapers? Actual print papers? They had a lovely article about it, and they said Sheena Josselyn, a sick kid scientist, politely declined an interview request from the Star because she had gone into labor. Still polite, true Canadian. This is a picture of my daughter a few months later, chewing on the copy of the magazine that the paper was published in. So that was our first foray into sort of manipulating memories in mice. But then, of course, we're now doing these wonderful new tricks to manipulate the activity of different cells in the brain. So this, you probably all have heard of algae and optogenetics, and it's a really cool way of using light to manipulate cells. So certain algae, they swim towards a light, and people have used this. Here's a picture of a bunch of algae. They shine light at one side, and the algae all swim on over there. So people have harnessed this to make these amazing tools that you can use to study brain science and just about every other type of science. So here we're using optogenetics. For us, it works like a light switch for the activity of neurons in the brain. So we use optogenetics, implant little fiber optics, little LED Christmas lights. Outside of the brain, we have a tube that delivers this light into the brains, and we can modify the activity of all these different cells. And of course, the Canadian Gairdner laureates were recognized. So we have Karl, Peter Hagerman, and Ed Boyden were recognized for optogenetics a few years ago. We'll sort of see if anything more comes of that. So this is a postdoc in my lab, Asim Rashid. As you can see, super hard working. So he said, you know, the studies that we've done before are pretty much dark ages. Why don't we use some optogenetic tricks to sort of play with memory and really do causal experiments in the brains of mice? So Asim did this very interesting experiment, and he took advantage of a cool tool developed by Karl. So this is a picture of, this is Karl, and this is me, and this is way back in 2016 at a Gordon Research Conference. And look at this, 2016, and Diceroth is already socially distancing from me. It's amazing, it's amazing. Always a forerunner. So what did he do? He made this really cool construct, and this construct expresses both an optogenetic actuator that increases excitability when you have blue light, so channel rhodopsin, and it also decreases excitability with red light. So this allows us to increase or decrease the excitability of the same population of cells simply by shining different colors of light. So if we shine a blue light, we're going to hit channel rhodopsin, we're going to increase the excitability of these cells. Then we can shine red light and decrease the excitability of the same population of cells. So this is a very cool experiment that Asim designed to do our erasure experiment all optically and reversibly. So what's he going to do? So he uses this viral vector that expresses channel rhodopsin and halar rhodopsin. He's going to infect a sparse population of random cells in the lateral amygdala of mice. He's then going to shine the blue light, and shining the blue light increases the activity of these cells, and we think that's the property that actually allocates them into an engram. So he's going to use the blue light. He's going to try and bias these cells particularly into the engram encoding this memory. We're going to train the mice, pair the tone with the foot shock, and then we're going to test the mice. But we're going to test them under two different conditions. We're going to test them normally once, and then we're going to test them when we turn off the activity of these cells that we've previously excited. So if the memory is indeed stored in these cells, if these cells are indeed part of the engram, we should expect to see pretty good levels of freezing in the same mouse without the red light, but decreased levels of freezing with the red light when we turn off the activity of these cells. And this is what we see. So this is a percentage of time that mice spend freezing to this tone. Without the red light, normally, the mouse is showing us good memory, so the mouse is freezing a lot. And then we turn on the red light, we just disrupt the activity of this small population of cells, and we see a disruption of freezing. So you could say, well, maybe if you just disrupt the activity of a small population of cells, you're going to see something. So as a control, he had another group of mice, but he didn't allocate these cells. So he didn't shine the blue light. These are just a random population of cells, similar to the cells that we killed previously. And when he turns off just a random small population of cells, nothing happens to the memory. So only when we turn off these cells we think are allocated really important in this memory trace do we see an effect. So that was a really nice all-optical way of doing this. So I've been talking a lot about memory encoding and how cells get allocated into a memory trace, but what about memory retrieval? So these were some really cool experiments done by Adam Ramsaran, and here he is looking very sleepy on some of our Zoom calls, Josselyn Lau and Jagrup. And they said, okay, we think in most of our experiments, what we're going to do, we're going to shine a blue light, we're going to allocate these cells into the memory trace. We're going to train mice, and then when we test the mice, we think that this tone, which is a really good cue that predicts the foot shock, we think that this tone actually reactivates these cells, and that's why a mouse freezes. So we think that this memory cue just reactivates this engram, this memory trace, and causes this mouse to remember and freeze. So we said, well, what if we take out the middle person? What if we don't play the tone, but instead just shine a light and artificially reactivate these cells? So these cells we think hold the memory, we're going to artificially activate them. We think that's what the tone does anyway, but we're going to do it optogenetically. So when we do that, we put mice in a novel context, nothing is happening. The mouse shows us very little freezing. We then turn on these cells we think are really important in the engram, storing the sphere memory, and a mouse is showing us freezing, even though we've never played the tone again. It's as if we're causing the mouse to artificially retrieve this memory. Now, people have asked, is this really the same thing as a natural memory retrieval? Is it not? If you artificially retrieve a memory, how is it different? And those are really important questions, and we're sort of looking at the answers to that. But I think it's really cool that you can sort of have a mouse bring something to mind just by artificially reactivating these cells. So using these and other techniques, we've been able to artificially select which neurons become part of an engram by increasing their excitability, so they become engram cells. We can artificially erase a memory. We do this both with a fear conditioning memory, and we also can do it with an appetitive memory, so it's not just fear. And to do this, we played around with a bunch of different things that mice like. So we tried first food to a hungry mouse, but we didn't get very good one-trial learning. So then we said, oh, chocolate milk. All people like chocolate, right? Even mice probably like chocolate. They do, but it wasn't enough to cause one-trial learning. So the best thing we could use that mice really liked that caused one-trial memory really, really robustly was cocaine. No judgment, no judgment. It's what's worked for us. So we could erase a cocaine memory in mice using these same different tricks. And we were also able to get mice to artificially recall a memory just by shining a light, reactivating these cells that we think are really important in a memory trace. So all these things are very, very cool. But they're all kind of artificial, right? We're optogenetics, we're shining light, we're trying all these different things, we're using viruses. But does this process of excitability-based allocation, which we think is really important for an engram, does it occur endogenously, or are we just sort of studying really odd things we can get a mouse to do? So this was the question that Chen Yan, who was a graduate student in my lab, asked. So what he wanted to do was to monitor the activity of different cells while a mouse was just learning and remembering something. And to do this, he made a little endoscope that he put inside the brains of mice, and his name is Chen, and he made an endoscope that we ended up calling a chendoscope. So this is a little miniature microphone. It has a cell phone camera and a little lens that we placed inside the brains of the mice. Here it is next to a toonie. We made it open so anyone can make their own. It costs a little bit more than a toonie, but not that much more than a toonie. So let me just show you our first iteration of how this works. There we go. So this is our very first time that we had it working. Here is our mouse here running around. It's got a chendoscope, and because we were leaking light, a bunch of tin foil. You can see the mouse running around the cage, but more importantly, you can see when each neuron is active. So it lights up because it's expressing a genetically encoded calcium indicator. So this tells us exactly when these neurons are active. So using this, we designed a very cool experiment. This is a picture of Chen. He moved on. He is now a research scientist at Google DeepMind. So he started my lab interested in molecular questions. He got into programming and a little bit of engineering, and that's the fun of science. You sort of go where your interests lie. So Chen is a really fun person, and the family really loves to go visit Chen, because if you know anything about the geography of London, you know that Google DeepMind is right across the street from King's Cross Station. Now this is my daughter Charlotte again, and she's a huge Harry Potter fan, so every time we go, we have to go to platform 9 3/4 to try and catch the train to Hogwarts. So this is what we did with the chendoscope. So we wanted to see whether neurons that are more active naturally are the ones that are recruited into an engram. So we did a really simple experiment. So we put mice in a cage for 5 minutes, and then we gave them a foot shock, and we tested their memory 24 hours later. And doing this, we defined which cells were engram cells, which cells were not engram cells, and then we just traced back their activity and said, are these cells endogenously more active at the time of training? Have we just sort of been amplifying this effect using optogenetics? So what we found was, yes, in this 5 minutes right before the mice receive a foot shock, the cells that go on to encode the memory are indeed more active than the cells that aren't important in encoding the memory. But we also noticed that these cells are more active in the mouse's home cage immediately beforehand, and even 3 hours beforehand, but not 24 hours beforehand. So this suggests to us that our thinking about how cells get allocated to an engram needed to be expanded a little bit. So it suggested to us that cells at any one time, different populations of cells are active, usually a small population of cells so that there's no epilepsy or anything, and that at any one time, should an event occur, these cells that are naturally more active than their counterparts are going to be the ones that encode this memory. So we think that there's fluctuations of activity throughout the day. The cells that happen to be more active at any particular time are the ones that are recruited into an engram. So we're following this up and looking at whether the connection between neurons is really important, and we're having a lot of fun using these really fun new tools to examine memories in mice. So with the aid of new tools, I'm going to say it's an amazing time to be a scientist, especially a neuroscientist. So we are quickly turning science fiction into science fact. So these are three movies about memory with a uniquely Canadian twist. So this is Eternal Sunshine of the Spotless Mind. Of course, Jim Carrey is from Scarborough, sort of on the outskirts of Toronto. This is Total Recall, a movie about memory. And the original movie with Arnold Schwarzenegger was filmed in Toronto, so Arnold. And this is Inception, a movie about implanting memories. No huge Canadian connection, but Elliot Page, who was in this movie, was born and raised on the east coast of Canada. So with the help of this amazing Canadiana, I think we are really figuring out how memory works. So with that, I just want to thank the wonderful people of my lab. Here we are on our annual selfie day. So the group stopped coming out for these group photos, but everybody likes taking a selfie. So we have an annual selfie day, and this is us pre-pandemic, and this is us during the pandemic. So we're not quite as happy looking, but we're still, managing to get through it. And with that, I just want to thank everybody and to say that I think it's a really amazing time to be a scientist. I think that we're sort of breaking down traditional barriers and that the sort of world is at your fingertips. So if you're at all interested, I say keep going in science. If you don't want to keep going in science, that's fine, but always be a promoter of science. I think science will save us all in the end. So thanks so much for your time and attention, and I'm happy to answer any questions.